Thanks to all of you for joining us for the 2023 June monthly seminar on physical genomics. My name is Benjamin Keene. I'm the Senior Program Administrator here at CPGE at Northwestern University. For those of you who are new to this seminar, this is CPG is a transdisciplinary research center at Northwestern that's primarily focused on studying the structure and function of the genome through a convergent approach of super resolution imaging, modeling, computational genomics, basic biology, artificial intelligence, with a mission to develop new strategies for the treatment of disease and the reversible engineering of living systems. You can learn more about us, our research, our training programs at physicalgenomics.northwestern.edu, or you can follow us on Twitter or Instagram. We're really excited to welcome as our speaker today, Dr. Bas van Steensel, who is research group leader at the Netherlands Cancer Institute. Dr. van Steensel received his PhD in 1995 from the University of Amsterdam, and he's held postdoc positions at the Rockefeller University and the Fred Hutchinson Cancer Research Center, studying various aspects of chromatin biology. He's an EMBO member, investigator of the OnCode Institute, and adjunct professor at Erasmus University, Rotterdam. His lab at NKI develops and applies new genomics technologies to elucidate principles of genome biology. So thank you so much, Dr. van Steensel, for sharing your work with us today. There'll be time for questions at the end of the talk, or if you just want to type them into chat, we can do that as well. So at this point, Dr. van Steensel, if you'd like to share your screen, I can cede the floor to you and we can begin. Thank you so much again.
Okay, well, thanks for having me. It's wonderful to contribute to this seminar series. Let me see where my presentation is here. And the laser pointer. So, does it look good?
Yes, we can see it. Thank you.
Let me see if I can just remove this little thing from my screen. Yeah. Okay, well, again, thanks for having me. I would like to talk about the work we do in our lab to probe the principles of genome organization and function. And I would like to start with the challenge of understanding mammalian gene regulation. Of course, we know a lot about gene regulation thanks to the work of an enormous community over the past decades. And the basic principles are fairly well understood. Some of the key players are promoters that drive gene expression, and they are in turn controlled by a wide range of enhancers. But what we still cannot do very well is make predictive models of gene expression. For example, predicting gene expression based on sequence, based on cell type, and really understand those underlying principles of the whole network of gene regulation. That's still a challenge, and that is in part because of the enormous numbers that we're facing. So there's roughly something like 50,000 promoters if we include some of the alternative promoters that many genes have. There may be a whopping 1 million enhancers, although we don't really know how many there are, but it seems like there's lots and lots of them that can control promoters. And then, of course, there's also the packaging of DNA into chromatin that affects the activity of promoters and enhancers. And that makes a very complex system to analyze. Now, what has been incredibly successful in the past decades is systematic deconstruction and the reconstruction of pieces of DNA to understand how they work. For example, promoters are now, the principles of promoters and their function is fairly well understood thanks to systematically taking apart the promoters, chop them up into small pieces, see how these individual pieces work, which ones contribute to the activity, then put them together again and see how they work as a whole. And the same for enhancers that we put together with promoters to see how they work together. That systematic reductionist approach has been incredibly successful. But given these enormous numbers of promoters and enhancers, we need to really scale up. And that is what I would like to talk about today, that the efforts of my lab to scale up these approaches to do systematic deconstruction and reconstruction of the elements and see how they work and how they work together. So I'm going to present three projects that we have in the lab. The first one is related to how regulatory elements and specifically enhancers and promoters work together. The second project that I would like to present is our efforts to understand how the localization of elements in the genome might affect their function. And the third topic that I would like to discuss is how chromatin organization, particularly the organization into very large domains, is controlled by DNA sequence. So let's get started with the first topic. How do regulatory elements work together? And in particular, the question, how do enhancers select their target promoter? We all know that enhancers can be located at a wide range of positions relative to their genes, and yet somehow they manage to find their target promoter. Sometimes the promoter is far away. Sometimes the enhancer is located between two genes, and somehow the enhancer really prefers one gene over the other. And there's also examples known of enhancers that seem to hop over specific genes to activate a more distal gene. So how does that work? Well, there's in the literature two models are prevalent, and one is related to the 3D organization, the idea that large loops might form that bring an enhancer close to the promoter and thereby help its activation in favor of another promoter that might not be involved in the loop. And the other model is, or explanation is that of intrinsic biochemical compatibility between enhancers and promoters. For example, certain factors need to bind to both the enhancer and the promoter so that they can actually sort of click together and thereby make a functional enhancer-promoter complex. Now, that's the model that I would like to evaluate a bit more. We have taken a very systematic approach to tackle this. This was work by Miguel Martinez-Ara in the lab, who developed a combinatorial enhancer-promoter assay. Basically, what he did is he took hundreds of enhancers and promoters and put them together in a reporter library in a combinatorial fashion. So he basically put PCR products of all the enhancers and promoters that he picked together in one tube, and then did random ligation to make pairs of enhancers and promoters, and those pairs he cloned into a barcoded reporter library. And then he sequenced everything to find out which enhancers and promoters were linked to each other and to which barcode they belong. And the barcode is in the transcription unit, so we can detect the activity then by counting barcodes in the RNA. As a control, or as a reference, he also made a promoter-only library, and that mix of libraries was transfected into cells, and in this case, mouse ES cells. And then basically, after a while, we isolate the RNA and we count the barcodes in the RNA. Now, then you have data activity measurements for the promoter only, but also for various combinations of enhancers with that promoter, various enhancers with that promoter. And then from that, we can simply calculate what we call a boost index, which is the fold activation of the combination of an E and a P compared to the P alone. Now, the loci that we chose were from mouse embryonic stem cells, 3 loci centered around 3 genes involved in pluripotency. From these loci, we chose in a range of 100 to 300 elements per locus, about 20 promoters each, combined with between 80 and 300 candidate enhancers, I should say, because most of these elements, it was not demonstrated that they were enhancers, but they had enhancer-like features, open chromatin, and often also K27 acetylation and other marks. So then we get, for each of these loci, we get a matrix of enhancer promoter combinations that for which we have these measurements of the boost index, and that's shown here. So on the x-axis you see the enhancers, on the y-axis the promoters, and the color indicates the boost index or the fold activation or the log2 fold activation. And in orange means that's activation. There's missing data. Note that that's indicated by the white squares. And we also see some signs of repression, although it's often very modest, and we still don't know really how to interpret that. So we have not done a lot of work on that. We have really focused on the activating parts. Now, if you eyeball this, you'll see that there's quite a lot of different patterns of activation. For example, here, this enhancer seems to activate many of the promoters, but here there's much more specificity, and here. So it seems like there's lots of different patterns visible. I'm just going to zoom in on a few of those patterns. So here we see a set of promoters from this locus here in combination with 3 different enhancers. Now, the first thing that you might notice is that this enhancer is apparently more potent because it activates many of the promoters more strongly than these 2 enhancers. But if you now look more specifically, more carefully at the patterns, you start to see differences. For example, if you look here, this promoter is about 5-fold more strongly activated by this enhancer E125 than this promoter. But if you look here, this enhancer activates both promoters to the same degree. Likewise, if we look here, enhancer 60 activates these 2 promoters roughly to the same degree. But here you can see that this enhancer activates this promoter much more than this promoter. So there's definitely a certain degree of specificity going on between enhancers and promoters. Now, we worried a lot about whether these things that we observed were just due to noise in the data, technical noise or biological noise between replicate experiments. We spent a lot of time doing the statistics on this. I won't bother you with the details. We've done now 2 different kinds of statistical approaches, and both say that for more than half the enhancers, the selectivity that we see for promoters is more than it can be explained by technical and biological noise. Now, there's been reported, particularly in Drosophila, that there might be some grouping of specificities and that housekeeping promoters have a preference for certain types of enhancers as opposed to non-housekeeping promoters. So we also analyzed it in our data. We grouped the data by the promoter type, housekeeping or non-housekeeping, and then looked at all pairwise enhancer interaction patterns. And the housekeeping promoters show a very strong similarity in their enhancer preferences. It's indicated by these Pearson correlation, the pairwise Pearson correlations. They're from almost all the pairs very high. The non-housekeeping promoters in red show a much broader distribution of preferences, indicating by this much broader distribution of Pearson correlations. And if we look at pairs of housekeeping versus non-housekeeping in gray, the correlations tend to be even lower, indicating that those types of promoters have different preferences. So we see clear evidence for compatibility between enhancers and promoters, and we wondered whether that has anything to do with the looping that has been observed between enhancers and promoters, and that can be mapped with high C technologies. So we wondered whether maybe this compatibility could perhaps help with the looping, right? It might make certain enhancers more sticky for certain promoters, or perhaps there was some kind of co-evolution of these mechanisms that when two an enhancer and a promoter find each other by looping, maybe they are also more compatible. So we compared our compatibility scores or boosting indices to contact frequencies as according to high C data from this source, and the result is quite clear. There is no relationship between the boost index and that observed over expected contact frequency according to high C technology or micro C technology. So it seems that these are really 2 independent layers of regulation. We have mechanisms of looping that help enhance find certain promoters, but we have also a separate mechanism, and that's this intrinsic compatibility. Now, this work was published, so you can find it in a paper that we published last year. From now on, I would like to talk about all unpublished work, and let's move to then the second topic, and that is the question how the location of elements affects their function. So let's think about this again. Very often it's observed that an enhancer that activates a certain promoter is located quite far away from that promoter. Now, if you or I would design a genome, you would probably do this differently. You would probably place that enhancer close to that promoter so that you have the most efficient interaction. But apparently nature doesn't do it like that. And so the question is, how important is that position of that enhancer or that promoter relative to the enhancer in the genome? So what you would like to do is take such an element, cut it out, and relocate it to different places in the area and see how that affects its function. Now, that's what we set out to do, and we wanted to do this in a very scalable manner. You could think of, well, let's do this by CRISPR, cut it out and insert it somewhere else. Well, that's going to be very tedious because CRISPR has a lot of efficiency problems, but also lots of, it tends to be quite mutagenic, so it would be quite scary to do that. So instead, we designed a hopping approach, making use of the Sleeping Beauty transposon. This is in part inspired by work by Francois Spitz several years ago, who pioneered this kind of work, but we wanted to really make this into a quite scalable strategy. So what we do is we take the Sleeping Beauty transposon and we inserted a simple recombination cassette with a positive-negative selection marker that allows us to easily recombine in enhancers or promoters of interest. That whole cassette, including the transposon arms, is inserted in a region of interest where we would like to hop the locus, the element around. Then we use the recombination cassette to swap in the element of interest, promoter or enhancer or something else. And then we mobilize the transposon, and it's known for Sleeping Beauty that if you activate it by adding transposase to the cells, then the element starts to hop around, but it preferentially hops around locally, and I'll show you quite some evidence that that's indeed the case. So then what we can do is isolate a pool of cells that has all these hopping events, and then in that pool of cells we have a very efficient way of mapping the integrations, and then the trick is then to link that location to a measure of gene expression or some other functional readout. And ultimately, we would like to do this in single cells, but right now we're doing this still in either isolated clones or in cell pools, as I'll show you. No, let's move to the data. So for a proof of principle and some exciting biology, we decided to focus first on the SOX2 locus in mouse ES cells. Now, SOX2 gene is one of the pluripotency genes in mouse ES cells. Interestingly, it's located in a large gene desert. It's pretty much the only active gene in the region, and it's strongly controlled by a very strong enhancer that's located more than 100 kb away, which is often called the SOX2 control region. If you delete this SCR, the expression of this gene goes down about tenfold. So it's really a very important regulatory element there. It's also known by high C and micro C data that this element physically interacts with the SOX2 gene. So what we asked is, well, what will happen given this distance between these two elements? What will happen if we move the gene, or actually we're going to do this only with a promoter, if we move it around, and how will it then respond to the different positions? Now, for this, I need to tell you a little bit more about the system. As I said, we're doing this in embryonic stem cells, but we're using an F1 hybrid, so the result of a cross between two distant mouse strains, the Castaneus, or actually species Castaneus. So one is from the mus Castaneus, and the other one is from mus musculus, from the 129 strain. And in this cell line, we inserted, or we tagged the two SOX2 alleles with GFP and mCherry so that we can monitor each individually. And we're making a lot of use of the SNPs that are present between these two mouse species so that we can always monitor the individual alleles. Now, so then we wanted to hop around a reporter that's driven by the SOX2 and promoter, and that we labeled in blue with the mTurk reporter, and that's captured in this what we call launchpad, from where we can hop this reporter around now. And this work was done by these fantastic 2 PhD students. So just to give you an impression of what such a hopping experiment looks like. So here you see the SOX2 gene, here you see that SCR, and this is the launch site. And this is the result of one single experiment where we hopped the element around and then we mapped the integration sites, and you can see that we get several hundreds of integration sites nicely distributed around the launch site, almost a Gaussian distribution. Maybe there's a little bit of a bias here towards that SCR, and we can talk about it if you like. But then what we can do is, because we have fluorescently tagged the SOX2 alleles and also the hopping reporter, we can then sort based on, and in this case, on that reporter, on that blue reporter, we sorted the cells by FACS sorting. Basically, after transposase transfection, we see a somewhat broadening of the reporter expression, and then we took 6 bins of expression and we sorted the cells, and then we asked what hopping events did we find in each of those bins. Okay, so the result is shown here. So here you see the 6 bins vertically. This is the unsorted control. Here's sort of the null distribution of hopping events. You can see that that's the same, the Gaussian distribution that I showed earlier. And then here you see the different bins of expression, and there's really more than 100-fold difference in expression across those that range, and where those hopped reporters landed in the locus. And what you can see is that with high expression, there's a clear bias in where they hop to. There's quite a few that stayed very close to where the gene was originally located, and many of them hop towards the SCR. That's maybe not so surprising because that's the enhancer, but clearly being close to the SCR really boosts the activity of the promoter. And then if we go down to low expression, you can see that all the lowly expressed ones are pretty much located outside this region between the gene and the enhancer. And there's this very sharp transition. As soon as you hop outside of this region between gene and enhancer, the expression is really low. So we were quite intrigued by that sharp drop-off, and we hypothesized that there are some mechanisms here at these 2 edges that may somehow prevent the reporter here from profiting from the activity of the SCR. So here, I'd like to focus here on what's happening on the left. And one possibility is that the SOX2 gene itself may actually sort of block any effect that the SCR might have further downstream, seen from the SCR then here on this side. So we said, well, let's take one of these clones located here, about 50 kb away from the gene, has a really low expression level, and let's now delete the endogenous gene, which is marked by M cherry, and see what the effect is on the expression of that reporter gene. So here's the experimental design. The reporter is located 50 kb to the left of the gene, and this is the SCR, which is located another 100 kb further down. And then we're going to delete either the gene itself or the gene together with the CTCF site. Actually, I'm not going to show you the data, but the result was pretty much identical. So here's the result. So this is a negative control. It's just a baseline of the FACS readout. This is that reporter located 50 kb upstream in the presence of the endogenous gene. But when we delete the endogenous gene, you get massive upregulation of that reporter. So apparently, the SOX2 gene itself forms a strong barrier for the activation of that reporter, upstream reporter, by the SCR. Now, here we need to talk about possible mechanisms, and one possible mechanism that many of you will know about is the loop extrusion model, which is now based on quite a lot of good evidence that the cohesin complex is able, which is a ring-shaped complex, which is able to pull DNA through its ring, and by a progressive pulling mechanism, that would eventually lead to the gene becoming close to the enhancer. Now, what we don't know yet is where that loop extrusion starts. It might start close to the SCR. It might also start close to the gene. That's still unclear. So we wondered, well, does loop extrusion start from near the gene, or does it start, and that seemed more likely in this case, does the loop extrusion start here from the SCR and reels in basically the DNA, and then the first gene that it encounters is this M-cherry labeled endogenous gene, and maybe this is why this further downstream or upstream, the way you look at it, turquoise labeled gene is not able to reach the SCR because it's just the second in line. So could that be the right model? So we wanted to figure out which way does loop extrusion happen, how important is loop extrusion, and in which way might it, in which direction might it work. So for this, we said, well, let's insert binding sites for CTCF. CTCF is a protein that's known to block loop extrusion, but it does so only in an orientation dependent manner. So by choosing the orientation of the binding site, you can block loop extrusion in one direction or in the other direction. So here, if loop extrusion starts from the SCR, then a CTCF site inserted here in this direction would have a strong effect on the activation, whereas if it would start mostly from here, then it would have a strong effect if it's oriented in this direction. So that's the experiment we wanted to do, but we also wanted to do it in a scalable manner, because you don't really know exactly where you should insert that CTCF site to have the most clear effect. That also depends on where loop extrusion really starts. So we leveraged our hopping approach to insert the CTCF site in many different locations. And here's the result of that. So what I'm showing you here is, so what we did was we hopped CTCF site around in the locus, and it also randomly lands in each of the two orientations. And then we sorted cells based on loss of expression of the endogenous SOX2 gene. And here you see the distribution of hopped CTCF sites that we obtain, and you can see that CTCF has the most potent effect, reducing effect on gene expression when it's located in between the gene and the SCR. So that is an argument in favor of loop extrusion playing a role, but now we can split up the data by orientation of the CTCF site, and that's shown here. And what is fairly clear is that orientation in the forward orientation has a more potent effect than the reverse orientation. So what does that mean? That means that loop extrusion is probably starting more from the side of the SCR and less from the side of the promoter, although it's not a day and night effect. There's probably also some loop extrusion starting here, but the majority seems to start here because that's indicated by the orientation of the CTCF site effect. Now, I should emphasize that really this loop extrusion seems to explain only part of the strong activation by the SCR. The effects that we see on the expression of the SOX2 gene are much less than what you see if you completely delete the SCR. So there's some other mechanism that also plays a role. Now, you may have wondered in the meantime, if that's true, if loop extrusion would start from there, then if you now insert this reporter here, then it would be the first in line. So does that mean that it now wins the competition against the M-cherry allele? Now, we looked at that. We had obtained quite a few clones that had the turquoise labeled reporter integrated here. And I'm showing you here the effect on expression on M-cherry. So here, all these dots here are integrations of that reporter relative to the endogenous M-cherry. And the bottom, this is the baseline activity of M-cherry when there's no integration of the reporter. And the bottom line is there's particularly for the guys that are located inserted in between the SCR and the endogenous gene, there's basically no effect on the expression of the endogenous gene. There's only a bit of an effect if the reporter inserts in the middle of the SCR, and that may be some kind of disruption of the SCR effect. It looks like it's not that simple. There's something else going on. There seems to be that the endogenous gene is somehow just much more powerful. It has a much better and intrinsic competitive edge compared to this reporter. Now, this reporter is driven by the same promoter, so it doesn't seem to be the promoter itself that explains it. It's something else in the gene. At this point, we don't know. The gene is tiny. The gene is only 2 kb or so. So it's very intriguing to think that there might be something in the gene itself that does it. But it could also be that there's still some kind of flanking elements here that help that gene to get its strongest potent, its strong potency compared to the reporter. And we're trying to figure this out further. Oh, that's the wrong one. Okay, then I would like to move to the third topic and project in my lab, and that relates to a slightly different question, but you'll see that the technology is quite similar. And that is the question, how does DNA sequence instruct chromatin organization? Now, I'm going to focus on one of my favorite chromatin types, and that's lamina-associated domains, or LADs. You may have heard of them. Basically, LADs are the chunks of heterochromatin that are associated with the nuclear lamina. We developed in the past ways of labeling them fluorescently, and you can see nicely interacting with the lamina. We can map them by DAM-ID, and some people can also do this by chip or other similar technologies. But basically, what you see here is the interaction of the genome with the lamina. This is about 10 megabases of in human fibroblasts, and you see these very large domains that interact with the lamina, alternating with large domains that don't. So these LADs, they're about 1,000 or a bit more than 1,000 in the genome. They're very big domains. They cover collectively about 40% of the genome. And what makes them really interesting is that they're late replicating. They seem to repress transcription. Well, they do repress transcription. If you insert reporter genes inside LADs, their activity is much lower than outside LADs. And also, they seem to have some effect on DNA repair, but I don't have time to discuss that. So, but what we still don't know is these very big domains, what in the DNA sequence dictates their interactions with the lamina? That's still largely unknown, or it's totally unknown. If you look at sequences in LADs, it's very disappointing. The only thing that you really see is that they're slightly more AT rich. They have some different transposable elements in there compared to interlad regions, but nobody really knows what in the sequence drives these interactions with the lamina. So that's what we would like to figure out. And for this, you would like to make perturbations, and we're starting very basic. What we would like to do is just cut out pieces, for example, delete large chunks of the DNA and see what the effect is. For example, if we make a cut like this, where we remove basically the border between the LAD and the interlad, what would happen? You could imagine that now the LAD spreads and invades, so to say, the region that is now joined to. But it could also be the other way around, that the interlad region is spreading and invading the LAD, and thereby partially detaching this piece of DNA from the lamina. Likewise, you would like to make inversions. So to make basically turn the piece of DNA around and see, well, again, the same question, what does it do to lamina interactions? Do you see spreading of the LAD? Do you spreading of the interlad, or maybe nothing happens? So those were the kind of experiments that we wanted to do in a systematic and ideally somewhat scalable manner. Now, you could say, well, do this by CRISPR editing, and you would have to do it pretty much one by one, because we're talking about megabases of DNA. So those are not very easy experiments. On top of that, we found that CRISPR editing inside LADs is not impossible, but the efficiency is low. So you would have to screen a lot of colonies to get the right editing event. And what we really worry about is secondary mutations of CRISPR editing, particularly if you make such deletions we've seen a lot that if you do this, that you get secondary mutations. Either you get the deletion is much larger than you want. Sometimes you get tandem arrays of the regions where you made your cuts. Sometimes we see that the deleted fragment is somewhere inserted somewhere else in the genome. All kinds of stuff that makes the interpretation of the data more difficult. Now, so Lise Dauban in the lab, a fantastic postdoc, developed a way that was independent of CRISPR editing, and that also is a bit more scalable. And that builds on the same principle of local hopping, again inspired by pioneering work from Francois Spitz about a decade ago, who showed that this is also possible in mice. But the essence is that we now again use the Sleeping Beauty transposon, but inside the only thing that we have in there basically is a LOXP recombination site. So what is it, about 25 nucleotides or so of a recombination sequence. And next to it, we have another LOXP site. And this cassette, we integrate in the locus of interest and then activate the local hopping. So then this LOXP site will hop around. And so you get now pairs of LOXP sites at various distances from each other. Now, if you now activate Cre recombinase in these cells, then if the two LOXP sites are in the same orientation, you get a deletion. And if you have them in antisense of an opposite orientation, you get an inversion. So that was the idea. And so Lise selected again in mouse ES cells a region that was particularly useful for this analysis, with one big strong LAD, about 2 megabases in size, and another somewhat weaker LAD, maybe consisting of 2 subLADs next door. And in that region, in between those 2 LADs, she inserted that launch site for the LOXP element. And then she mobilized the site and she mapped the integrations. And she was quite disappointed because this is the interlad region in white, and in blue you see the 2 flanking LADs, because she barely got any hopping events from the launch site into the LAD, and that was what she wanted. She wanted to make disruptions inside the LAD, or in particular of that junction between the interlad and the LAD. So she was quite disappointed because she got very few of these hopping events. And she also saw that in 2 different loci where she did similar experiments. Again, the majority of hopping events seem to be constrained to the interlad region if you start from an interlad region. Now, maybe in hindsight, that makes sense because LADs and interlads are spatially separated from each other in the nucleus. And so maybe if a transposon really hops by excising itself out and then basically inserting into the first piece of DNA that it encounters, then the chance of hopping from an interlad to a LAD might be quite small. But Lise realized that, well, she identified a few clones where she had a hopping event inside the LAD, and then what she did was simply remobilize the transposon from there. And that worked quite well. And then you see that she gets lots of hopping events inside the LAD, but not many outside of the LAD. So again, confirming that there's this spatial separation that constrains these hopping events. But she was happy because now she has a lot of hopping events inside the big LAD and the small LAD. So she isolated clones that had those hopping events, and they're listed here. The ones that she focused on are listed as shown here. So here inside the big LAD and here inside the small LAD, and the orientation of the LOXP site dictates whether you can generate an inversion or a deletion. Now I'm going to show you some of the data, not all of it, because Lise did an enormous number of experiments like this. But basically, the experiment the next thing was, does that recombination now work? So how efficient will it be? So Lise transfected in the Cre recombinase and isolated clones and used PCR to map inversions and deletions, or identify clones with inversions and deletions. And my biggest worry was the efficiency of this process, that the recombination efficiency would be too low. But actually, it's really not that bad. On average, the efficiency is in the range of 10%, even with recombination sites that are about 2 megabases apart from each other. I find this amazing that you have these two little elements of 25 nucleotides or something that are separated by 2 megabases, and yet they find each other in a week time with an efficiency that's in the range of 10%. It blows me away that that's possible, and there's maybe some really interesting biology there to figure out why that is so efficient. So Lise also did these experiments in F1 hybrid mouse ES cells with a CAST and a 129 allele, because she makes her rearrangements only in one allele, and she uses the other allele all the time as a control, as a reference, to make sure that the effects are really in cis and not due to some overall change in the cells. We mapped the lamina interactions with our PA DMRD technology. I don't have time to discuss it, but it's reported here. It's a very efficient. It's sort of a hybrid between cut and run and our DMRD technology. That's very efficient and very yeah, doesn't take a lot of time either. Now I'm going to show you a couple of data slides, let me take you through what these slides, what this data representation looks like. It's always consisting of 3 panels. Here at the bottom, you see just the location of the genes and their expression level. Here in the middle, you see the lamina interaction tracks. In blue, the control cells, and in red, the experimental cells. And in this case, it's a deletion strain where this region has been deleted, and so there's no data, of course, no lamina interactions in this region. And you can see here the effects on the lamina interaction in the flanking regions. And if there's an increase in lamina interactions, the shade is red. If there's a decrease in lamina interactions compared to the control, it's a blue shading. And then here on top, you see what we in the lab often call tornado plots. It's sort of a statistical approach to gauge whether the effect that we see is more than you might expect based on just noise or random fluctuations in the data by comparing it to the rest of the genome. And this analysis is done on many different scales, from about 20 kb to about a megabase. And you get also an idea of how big is the region that detaches or attaches relative to the control. So let's focus on a couple of the deletions that Lise made that make different deletion sizes of this big lad. So let's first look at one of them. This is a relatively small deletion that affects only part of cuts only a part of the lad. And what you can see is that now the flanking region, which is now joined to this lad, starts to show an increase in lamina interactions. And there's maybe a little bit of detachment of the lad here, but no, I wouldn't say really. Actually, the tornado plot also says that that's not really the case. So you get some kind of rearrangement where you get some invasion of the lad into the neighboring interlad. Now, if we make larger deletions, that effect becomes a bit stronger. And only if we make much larger deletions, we start to see that the remainder of the lad starts to dissociate from the lamina. Here now you see the blue tornado here. So here's an overview of the tornado plots for 6 different deletions sorted by size. And you can see that on the left, the effect is more or less the same, but on the right, only if you make a large enough deletion, then the remainder of the lad is becoming detached. And there's a very sharp transition here between this deletion and this deletion of there's a region of about 150 kilobases or so. And so we wondered, maybe this is a critical element in the lad. Maybe this is what we refer to as the lad nucleating region when we saw these data. So is that how important is that element? Well, for this, Lise made actually a CRISPR deletion just to really make a clean deletion. And maybe disappointing or not, it depends on your view. She found that deleting this region just by itself, this putative nucleating region, had no effect whatsoever on the interactions of this lad. There's some weird stuff happening here and here far away that we don't understand, but in the proximity of this region, there was no effect. So apparently it's not essential for this lad to interact with the lamina. Now, is it perhaps sufficient to do such a thing? For this, Lise wanted to isolate this region in an interlad region or between 2 interlad regions that was a more complicated experiment that involved a combination of an inversion and a deletion. And let's not worry too much about how she did it, but this is the region that she was interested in, this putative nucleating region. It's now positioned here next to an interlade, and on this side also next to an interlade. What you can see is that compared to its original strength, it has dropped quite a bit. So it really helps, it really needs its environment, its normal lad environment, to be very strongly associated with the lamina, but still it's still higher than its flanking regions. And now you can see that the region that is now joined to it, an interlade region, actually shows an increased lamina interactions. So this region does seem to be to some extent able to enforce lamina interactions, but it really does a much better job if it's flanked by other elements that apparently help with it. So take-home message is that these lamina interactions are cooperatively and redundantly controlled, and so that makes it also quite hard to analyze in much more detail. Basically, we've seen this 150 kb element that does play a role, but it doesn't do it all by itself. And yeah, that's what I said here. Some of the LAD subregions contribute more to these lamina interactions than others. Now, one last little bit then is what does all this do to gene regulation? These changes in lamina interactions that we can enforce by making these rearrangements. Well, we did RNA-seq on this, allele-specific RNA-seq on this, and I'm just going to show you the effects on the affected allele. The other allele, actually, we looked at, and there's no such effects visible. So I'm just going to show you the effect of this deletion here, which had this increase in lamina interactions of this flanking interlade region. And if we look at the gene expression in two different clones, we see nicely that two genes that are located just close to this edge that now show an increased lamina interaction, they show a drop in expression. So that suggests that lamina interactions suppress gene activity, but we cannot completely rule out that in this deletion that we made, that there's somehow here an enhancer that is necessary for full activity of the genes that are located here. We cannot rule that out. But the most parsimonious interpretation seems to be that lamina interactions have a repressive effect, and that fits with a lot of other data that we and others have generated. So activity of some genes is affected, but we see that not all genes are affected. And so there's still a puzzle to be solved why some genes are more sensitive to this than others. So to summarize, from the technology perspective, I present to you some platforms that we've set up for a systematic and more scalable deconstruction and reconstruction of genomic elements and their interplay. A combinatorial massive parallel reporter assay to look at the enhancer-promoter interplay. We are now working on a follow-up where we look at enhancer-promoter interplay, so to see how enhancers work together to activate promoters. I showed you how we can now hop regulatory elements around in an efficient way to hundreds of sites in a single experiment. Promoters, boundary elements, we're working on hopping around enhancers now. And the same tool can also be used to hop around recombination sites to more efficiently generate deletion series and inversions, etc. So with this, I would like to end, but not before thanking a fantastic lab. I've already highlighted the people who did most of the work that I presented today, but there's a lot of contributions from others in the lab and a lot of discussion, etc. Groundwork done, amazing pioneering work done by these people, lab alumni. And I should say that this hopping project independently, Luca Giugetti's lab developed a similar kind of tool. And when we figured this out, we got in touch and we agreed that instead of competing, we would collaborate. And so we have now joint lab meetings about every 3 months or so to update each other about all the newest findings and newest technology improvements. And that is so much better than competing and so much more fun. And we have a set of wonderful core facilities in our institute that made this work possible. So that's it. Thank you for your attention. I'm happy to take questions. And I guess I'll stop sharing here.
you so much. That was fantastic. Like I said, if anyone has questions, feel free to just unmute yourself, or if you'd like to type them in the chat, that works too. So we can open the floor if anyone has anything.
I can get us started. Boss, this is Alistair. This was fairly beautiful. It's fabulous. A quick question on the last part. I was curious what cell type you've looked in. And as you know, a lot of in the MES cells, there's not a lot of, or the lab interlab difference is not quite as strong as in the highly differentiated cells. And I'm curious if there's insights into like how that regulates.
All these experiments were done in mouse ES cells. Yeah, you're saying the LADs are not as strong. They're a little bit weaker, that's true, than the more differentiated cells. But yeah, I'm not sure that the reason why we're using these mouse ES cells is that they're F1 hybrids, and so we can track the two alleles very efficiently. And there's not many other systems that you could use for that. So I mean, we could differentiate them maybe to neural precursor cells or some other cell type, but we haven't done that yet. Yeah.
That's great.
Can I ask a question? Dr. van Steensel, beautiful work as usual. Raj Jain at UPenn. I had a question about the third part. The deletion are incredibly elegant experiments. I was wondering, do you have a sense of the expansion of the LAD in those examples you showed, the Lamina context, why it stops at that particular spot that your data showed it stopped at? Is there a unique, series of genomic elements or something like that? I was just wondering if you looked into
Yeah, it's a good question. So the spreading is in the range of 100 to 150 kb, and it seems to stop at a very active gene. Which is maybe not so surprising because there's a lot of evidence now for antagonism between gene expression and lamina interactions, mutual antagonism it seems to be. So that probably is the explanation. We're still, we wanted to do some experiments to see if we could kill that gene, the activity of that gene, and see if then it spreads further, but those experiments have not been technically not successful yet.
Thanks so much.
Hi, Beth. This is Lu Bai here. So the interaction with laminin was shown to be at least correlated with H3K9 trimethyl. So just wondering if you guys have looked at that. And basically in the end, it could be that you're just looking for some sequence elements that initiate that repressive mark, right?
Yeah. We have now mapped the K9, I think it's particularly K9 trimethylation in these regions, and indeed the LAD has more K9 trimethylation. The interesting thing is that spreading that we see for lamina interaction, right, if we make that deletion, does not seem to be the case for the K9 trimethylation. So we have uncoupling there, basically. It's actually remarkable that the K9 trimethylation stays exactly at that original at that border, or it doesn't invade or doesn't spread where the lamina interaction seems to spread. Now it's a bit preliminary, so we need to analyze this a bit more. But that's the initial view that we have, and so there seems to be an uncoupling there.
That's interesting. Yeah.